Chapter 3. Intake. The Earth's material composition and crustal structure remained relatively stable from its formation until the last few billion years. On the primitive Earth, water absorbed most of the excess energy from the surface, resulting in only slight fluctuations in the total energy of the Earth. Energy conversion and flow occurred within a small range of fluctuation between the crustal layer and the atmosphere, regardless of volcanic eruptions, earthquakes caused by crustal movements, or glacial periods. The solar system's sun and other planets are generally stable, and the energy received by Earth does not usually fluctuate significantly. Thus, it can be asserted that the vast majority of energy on Earth is in a fixed state. The energy that can be constantly converted and flows above the surface is mainly supplied by the sun, and a small portion of it is in free or semi-free form. All living organisms on Earth derive energy directly or indirectly from these sources, although the strategy and effectiveness of their intake vary from species to species. Selection of energy locus. With the exception of sunlight, the energy available on Earth for direct or indirect biological use is generally at the chemical level of molecules and macromolecules. This type of energy is relatively weak and discrete, which is suitable for biological uptake, transformation, and utilization, and is referred to as discrete energy in this book. Discrete energy on Earth is available at 3 levels. The first level includes non-biological material energy such as sunlight, inorganic compounds, e.g., carbon dioxide, water, small organic molecules, oxygen, nitrogen, hydrogen, and ionic minerals in the soil. The second level is secondary energy, which is the chemical energy contained in green plants, autotrophic and heterotrophic organisms. The third and final level is tertiary energy, which is the chemical energy found in animals. During evolution, a species must select one or more appropriate energy sources by choosing from a single or multiple levels of energy loci. The specific energy sub-loci and their sources are selected based on the level of energy abundance within their respective environments. Next, the corresponding biochemical reaction types, enzyme synthesis mechanisms, and unique characteristics supporting them are selected based on the specific energy source's characteristics to develop an intake capacity with certain advantages. All organisms on Earth are descended from a common ancestor as organic life. Thus, there will be several overlaps among the energy loci and subloci, and they will not be entirely distinct. When the interval of differentiation is shorter during the evolutionary process, the energy locus strategies of the two species will be more similar. The energy locus and its source of plants are usually light energy, water, carbon dioxide, and minerals, etc. Energy is primarily produced through photosynthesis. The internal chloroplast plays a crucial role in this process. Through the aid of sunlight, chloroplasts convert carbon dioxide entering the leaf through the stomata and water absorbed by the roots into glucose, while releasing oxygen in the process. However, some green algae have developed an unusual energy loci strategy that is distinct from that of other green algae. The research team of Professor Olaf Cruz at the University of Bielefeld has discovered for the first time that rhodophyta can photosynthesize and extract secondary energy from other plants. By including cellulose as a source of chemical energy in their energy loci, it refutes the mistaken belief that plants only absorb primary energy. Certain plants have evolved the ability to obtain tertiary energy, which makes them insectivorous. These plants have unique structures that enable them to capture insects or other small animals. They break down the captured prey by utilizing digestive enzymes, bacteria, or both to digest the tiny insects and absorb their nutrients. The captured prey is digested by the insectivorous plants in a similar way to animal digestion. The plants absorb the end products of the decomposition, particularly nitrogen compounds and salts. Carnivorous plants can carry out photosynthesis and digest animal proteins. They can adapt to challenging environments in specific situations due to the expansion of their energy reserves. The hot springs on the seafloor contain minerals and hydrogen sulfide, which serve as the primary power source for sulfur-oxidizing bacteria and are essential for survival. Heat-tolerant sulfur-oxidizing bacteria form the foundation of the food chain in the volcanic ecosystem. They also provide energy for plankton, which is then consumed by crustaceans such as shrimp and clams. The ecosystem surrounding the volcano relies on hydrogen sulfide for survival instead of the energy produced by photosynthesis. There is no shortage of primary energy on Earth. Since primary energy is so abundant, all organisms occupy some of the almost free energy loci without fear. Ants, wolves, and fir trees don't worry about other creatures or plants taking their oxygen, water, or carbon dioxide. In a dense forest, trees grow their branches and leaves as close as possible to where they can easily absorb sunlight. Seedlings, which are closer to the ground, have limited access to sunlight and can only use the available light spots for photosynthesis, resulting in slow growth. Competition for secondary and tertiary energy loci is fierce and can lead to hunger and danger to life. The quality of the energy loci determines the intensity of competition. Each species may become an energy locus for other species as they compete for its energy locus or loci. Predation and anti-predation interactions are ongoing. In the case of plants, their energy loci are targeted by other organisms, including herbivores, omnivores, and microbes, with herbivorous insects being the most prevalent. They consume plants and utilize the chemical energy that plants derive from sunlight to produce energy that is suitable for their needs. However, due to intense competition, they do not consume the entire plant as their energy loci. Instead, they select specific energy sub-loci to fulfill their genetic mission. For instance, caterpillars and grasshoppers consume plant leaves, while aphids and ticks extract plant sap. Bees collect pollen and nectar from plants, and beetles and flies feed on plant fruits, sometimes damaging their root systems. Even the trunks of large trees can be consumed by the larvae of wood-boring insects. Phytophagous pests vary in their degree of specialization in choosing sources of energy. They can be categorized as A. Monophagous pests, which parasitize only one type of plant. B. Oligophagous pests, which feed on plants of only one family and closely related families. C. Polyphagous pests feed on plants from multiple plant families. Pests that are oligophagous or polyphagous often exhibit preference in the variety of plants on which they can feed. Specialized pests may exhibit selection for different plant species within the family, which they typically consume. To summarize, the choice of where to locate energy and where to obtain it is linked to the abundance of the supply and the level of competition. In an ecological community, a significant decline in the energy supply at a particular location will trigger an increase in competition among organisms that depend on it. At this stage, the creatures that feed on the same energy source may start fighting each other, leading to a significant decrease in the number of species and populations. When certain omnivorous species adjust their energy structure or expand their energy loci, it is possible for the effects of competition and aggression to extend to nearby energy loci. Plants make it difficult for phytophagous pests to take away the secondary energy loci they synthesize, even at the expense of their own health and lives. To defend themselves, some plants produce toxic substances that are harmful to pests even if their gnawing does not benefit them. These plants use toxins as chemical weapons and store insecticidal proteins or other metabolites to deter pests. If a pest ingests the toxins, it becomes poisoned and suffers from indigestion, thus reducing the pest's impact on the entire species. Some plants emit odors that are repellent to pests, such as lantana. Lantana's branches, leaves, and flowers emit an odor that flies hate, making it a highly effective fly repellent. Several other plants in the plant world resemble lantana, including well-known ones like Murraya, Telosma, and Pelargonium graveolens. Over 2,400 plants worldwide were discovered to have toxic energy that can control pests. Artificial chemical pesticides also contain toxic energy. Healthy plants maintain a balance between input and output of energy, which allows them to take in their own energy at a high rate. According to Francis Suposo, a French biologist and author of Plant Health and Damage, pests growing on healthy plants will starve due to the theory of feeding symbiosis. Suposo argues that pests require an oversupply of hydrolyzed nutrients from the plant sap to maintain normal growth and reproduction on the host plant. Pests are unable to absorb proteins directly as they lack the necessary hydrolytic proteases in their bodies. Suposo's theory suggests that when a plant is healthy, the body utilizes numerous amino acids, sugars, and minerals to create new proteins, leaving minimal nutrients in the cytosol. Currently, the pests that rely on cytosol as the locus and source of energy cannot obtain sufficient energy to fulfill their requirements. Humans possess an unmatched advantage over other creatures when it comes to occupying the energy locus, mainly through early adoption of controlling fire. Fire is the only energy locus occupied by humans, providing them with a high capacity to absorb and utilize energy. Prior to the discovery of cooking with fire, humans could not use branches, mature leaves, extended roots, or tubers as energy sources, as they relied on simple sugars and carbohydrates found in plants like seeds, flowers, and juicy fruits. Cooking over a fire can help eliminate toxins like cyanogenic glycosides found in some sources, like flaxseed and tree potatoes. In addition, cooking meat makes it more easily chewable and digestible, leading to lower energy usage in the body than when consuming raw meat. Simultaneously, cooking eradicates hazardous biological agents like parasites and germs in food. This process can be equal to eliminating unseen rivals in the same food energy loci. Intake efficiency. Individuals with basic knowledge of nutrition understand that appropriate nutrient intake involves not only a well-balanced nutrient ratio, but also consuming greater quantities of higher quality foods. The term eating high-quality food refers to the energy intake efficiency. Studies on ecology commonly focus only on the food chain's flow inside the ecosystem. The energy flow in the food chain is unidirectional and gradually decreases. Unidirectional flow implies that the food energy can only move through the food chain from the first to the second level, and subsequently to the following levels in an ecosystem. The direction of energy flow cannot reverse since it abides by the trophic system of organisms that have developed for ages. Carnivores feed on herbivores, but the reverse does not happen. The progressive decline of energy flow implies that the full amount of energy exiting one level of nutrition inexorably decreases along the food chain, instead of all being transferred to the next level. Yale University ecologist Raymond Lindemann states that the energy transfer efficiency along the food chain averages between 10% and 20%. The energy usage of the lower trophic level is only 10% to 20% of that used in the upper trophic level. Plants are the primary source of organic energy in the food chain, and they convert inorganic matter to organic matter mainly through photosynthesis. Nonetheless, their energy conversion efficiency is only about 6%. However, studying only the transfer or conversion efficiency of light energy or food is not enough. All organisms can obtain energy from the first level, as proposed by the 3 levels of energy. Plants mostly obtain energy from light and inorganic matter, while animals get it primarily from organic matter. However, animals also require taking in small amounts of energy from light and inorganic matter. For instance, the lack of sunlight and the inability to utilize light energy can lead to various problems in the human body. Rickets, chondromalacia, and other diseases may occur because sunlight does not convert vitamin D3, which promotes calcium absorption. B. Susceptibility to colds can increase because the immune system may require light energy. C. Ultraviolet rays from the sun can have a bactericidal effect and break down toxic energy on the skin, leading to susceptibility to skin diseases. D. Light energy may contribute to the secretion of dopamine, synthesis of enzymes associated with brain activity, and conversion of neurotransmitters associated with sleep, which can lead to susceptibility to depression. E. Blood circulation and metabolism may decrease. The second law of thermodynamics governs energy transfer and transformation. During energy transfer and transformation, there is a portion of energy that can continue to be transferred and do work, free energy, but there is always a portion of energy that can't continue to be transferred or used. This energy is dissipated as heat and increases entropy. When energy is transferred between organisms, a substantial portion of the energy is converted to heat and dissipated. Some of the energy is used to synthesize new tissues to repair aging, dead, or damaged cells, while some of the energy gets stored. When an organism receives energy from food, it mainly converts it into heat needed for life activities. Most energy is converted to heat and lost when transferring between organisms, which thermodynamically explains the pyramidal shape of the energy relationship in an ecosystem and why there are no more than 6 trophic levels in a food chain. All organisms on Earth share a common ancestor, resulting in the inheritance of primary energy forms and structures despite long periods of differentiation and evolution. The energy forms and structures in current organisms are similar, resulting in a relatively high absorption rate of chemical energy. Carbohydrates have the highest absorption rate of about 97 to 98% in mixed human diets, followed by fat at about 95%, and protein with the lowest rate at about 92%. Nutrients are absorbed and digested at varying rates, and the rate at which an ingredient is absorbed varies depending on its source. For instance, the absorbability and digestibility of proteins differ significantly depending on their sources. For instance, the digestibility of legume protein is approximately 78%, while that of animal protein is generally much higher at 97%. This variation is connected to the species' energy form and structure evolution. The greater the time period between the divergence of 2 species on the evolutionary tree, the larger the genetic discrepancies determining the energy form and structure of each species. When one organism of a species is ingested as food by another species, the difference in energy utilization increases with the genetic distance between them. The type, quantity, and composition of essential amino acids in a food protein are critical to the protein utilization in the human body. To put it differently, the greater the similarity between the amino acid patterns in dietary proteins and those in the human body, the more efficiently the body will utilize those proteins. Thus, animal proteins are digested and absorbed more efficiently in non-human animals compared to plant proteins. Ingesting mammalian meat may result in a higher overall utilization rate in the human body compared to non-mammalian meat. Breathing is a physiological process required for the basic survival of any organism. Animals need oxygen in the air to carry out cellular metabolism and produce energy, typically using it to convert ingested food starch into glucose. As a general rule, the air we breathe contains around 21% oxygen, while each exhaled breath from a human body contains 13 to 16% oxygen. Therefore, the human body only absorbs 24 to 38% of the oxygen present in each breath, a relatively low absorption rate compared to that of animal or plant food sources. If the amount of blood is reduced or the flow rate is slowed, it can lead to insufficient oxygen supply to the cells. The body can experience tissue toxicity, cell damage, vitamin deficiency, and other oxygen utilization disorders at the tissue and cell level. At this stage, the body's ability to absorb oxygen is further diminished. Oxygen therapy is commonly used in medicine to enhance the oxygen content of inhaled air and improve oxygen energy utilization. Oxygen therapy has the potential to treat several diseases by rectifying hypoxia, elevating arterial partial pressure of oxygen and oxygen saturation, improving tissue hypoxia, promoting metabolism for maintaining the body's vital activities, and so on. The human body's oxygen absorption efficiency is a complex issue that's interrelated with the intensity of life activities and energy sources, which require careful consideration. No compelling evidence exists regarding the human body's absorption efficiency of primary energy sources, such as light energy, nitrogen, and minerals. Conversely, substantial advancements have been made in studies regarding the conversion of nutrients to energy when combined with oxygen. Most of the body's energy comes from sugars, fats, and proteins found in food. These organic macromolecules possess concealed chemical energy, which can be unleashed exclusively via aerobic respiration. Nonetheless, the energy from nutrients in food cannot directly be utilized by the body's tissue cells, thereby requiring the production of an intermediate compound, ATP, adenosine triphosphate, to provide energy for various essential activities in the body. The process of producing ATP is generally split into two main stages. Initially, sugars, fats, and proteins are consumed as substrates, with sugars being preferentially converted to pyruvate through the process of glycolysis in order of effectiveness. Pyruvate is then transported into the mitochondria, also known as the powerhouse of the cell, and undergoes the tricarboxylic acid cycle until it is fully oxidized. Throughout this process, the energy created through substrate-level phosphorylation, as well as the reduced forms of nicotinamide adenine dinucleotide, NADH, and flavin adenine dinucleotide, FADH2, is deposited in the form of ATP. An example of aerobic respiration with sugars can be simplified as follows: reaction conditions are omitted. C6H1206S plus 6O2G, right pointing arrow 6 Colorado 2G plus 6H2O L plus energy. Glucose is broken down aerobically through a series of chemical reactions. To complete aerobic catabolism, each glucose molecule requires the consumption of 2 ATP, leading to the production of 30 to 38 ATP. Of the energy produced by this catabolism, approximately 40% is stored as ATP, and the remaining 60% is dissipated as heat. This heat is responsible for maintaining body temperature, reaction conditions, and ATP reuse. Plants' absorption of light energy can be highly efficient. Photosynthesis in plants initiates with the light reaction, which produces ATP to fuel the subsequent carbon reaction. The photoreaction stage of ATP production renders chloroplasts entirely self-sufficient in their ATP consumption. Hence, chloroplasts neither supply ATP for external physiological processes nor absorb and utilize ATP from respiration. Water, which exists in diverse states in different life forms, plays an indispensable role in enhancing energy intake efficiency in organisms. Primitive life is comprised of three main elements: water, inorganic ions, and organic molecules. Living organisms are water-containing systems because of water's high specific heat and heat of vaporization, which make it the best thermoregulator for organisms. Bodily fluids under normal physiological conditions carry nutrients and waste to specific parts to fulfill important carrier functions within an organism. Water, being an excellent solvent, provides a suitable medium environment for life. The nature of biochemical reactions and reaction rates are determined by pH, ionic species, and ionic strength. Water participates actively in crucial reactions including photosynthesis and glycolysis. Figuratively speaking, water acts as an imperceptible energy blade, which easily dissects molecules or directly interacts with them. Our common predecessors mastered the use of this energy blade, applying it ingeniously to shape diverse energy forms and structures in their favor following a sudden enlightenment billions of years ago. One should never overlook the importance of symbiotic bacteria in the intestinal, digestive tract of multicellular organisms. The typically omnivorous human relies on symbiotic bacteria as essential helpers for efficient food ingestion. Of all creatures, humans have the most heterogeneous diet, which includes meat, vegetables, grains, fruits, and everything in between. Although eating food is critical, digesting, absorbing, and utilizing the food are equally important. Disregarding the efficiency of the input is a kind of blasphemy to life itself. The intestine is the most important digestive organ in the human body. The intestine, despite its multitasking abilities, cannot perform the challenging tasks of digestion and absorption alone, especially with the wide variety of foods consumed. Perhaps as a result, the gut offers a suitable and relatively anaerobic environment for symbiotic bacteria. Probiotics can reciprocate the favor, as they are skillful in breaking down complex fibers and polysaccharides, delivering decomposed glucose, vitamins, fats, and trace elements to the intestine for absorption by the body, much like paying rent. Both parties benefit each other by taking what they need. Competitive intake capacity. The energy locus occupied by a species and the selected intake efficiency are not permanent or unchanging, nor do they necessarily preclude new competitors. Thus, every species must also choose an appropriate strategy for energy intake capacity to gain a competitive edge, thereby maximizing survival and reproduction. If too many species in a given area evolve towards the same intake strategy and capacity, they may both lose and be eliminated by natural selection. Different species with an identical or similar energy locus must further refine and differentiate their feeding strategies, that is, evolving unique feeding capabilities and corresponding biochemical response mechanisms in their respective sublocus based on the original energy locus. For plants growing in rainforests, selecting the appropriate spatial level and intake capacity is the optimal strategy. Tropical rainforests typically exhibit vertical stratification of at least 5 layers, including the forest canopy layer, the understory layer, the shrub layer, and the soil surface layer. The forest canopy layer is the domain of those plants with the most incredible growth rate and transport capacity. The next layer is the canopy layer, occupied by plants with extensive leaf areas and high levels of photosynthetic activity. The thick, extensive branches and leaves of canopy plants absorb energy reserves and limit the growth of forest canopy seedlings. The understory leaf layer in the rainforest provides a habitat for smaller species, young plants, vines, and other trees that can absorb as little as 0.5% to 5% of the unblocked light. These plants have limited energy available to them and rely mainly on their roots to absorb nutrients from the ground. They must wait for a neighboring large tree to fall before they can receive more light. Shade-tolerant plants, including those with insectivorous and epiphytic abilities, occupy the shrub layer and ground surface. Animals have developed a diverse range of energy intake capabilities, with some evolving specialized predatory organs, such as claws, mouth needles, tongues, chelicerae, venom, sticky filaments, and suckers. The modification of some receptors to be more aggressive might have facilitated the evolution of certain predatory organs. Chelicerates, for instance, may have developed predatory organs from tentacles through evolution. Anomalocaris is the most ferocious and aggressive predator among the Chengjong fauna's significant arthropods. An anomalocaris predatory appendage comprises as many as 14 limbs, which are tightly gripped by 2 powerful forelimbs to capture prey. Anomalocaris' massive mouth has a maximum diameter of 25 centimeters, with a circular set of external teeth on its outer rim and 8 internal rows of teeth in the oropharynx. When Anomalocaris swallows, the external teeth's free end moves away from the mouth, gradually opening it. When prey enters the mouth, the free end moves back to its original position, closing the mouth gradually, while the serrated edge grasps the prey and transfers it to the mouth. The primary predatory organ of the centipede is its jaw, which contains venom glands. After the prey is paralyzed due to the venom of the centipede and loses its resistance, the centipede uses its large jaws to tear the chitinous outer skin and cut the muscle and visceral tissues of the prey's soft parts. Although most animals do not require specific predatory organs, the primary function of all such organs is to help them ingest food efficiently. For instance, the shape and size of bird beaks closely relate to their specific feeding habits. Birds with short, straight beaks primarily eat insects. Birds with beaks that have solid and curved hooks at the end feed mostly on birds and small animals. Birds with long, straight beaks feed mainly on fish and shrimp, while those with flat beaks are primarily omnivorous. Organs involved in food processing should be modified to accommodate intake, including mouthparts, saliva, teeth, and stomach, among others. Invertebrates commonly swallow food in its entirety. On the other hand, arthropods utilize their head appendages to shred food before ingestion. Reptiles possess teeth and a powerful bite, primarily utilized to grip and capture food. Unlike reptiles, birds lack teeth and usually consume food either fully or after breaking it with their talons before storing it in their crop where it softens. The majority of mammals have teeth and powerful jaws to break down food. While eating, the majority of needle-sucking insects release both gelatinous and watery saliva. Prior to feeding, their gelatinous saliva is discharged to form a barrier around the protective oral needle, preventing damage to plant tissue and enabling them to feed for extended periods from the plant's phloem sap. Their watery saliva consists of pectinase, cellulase, polyphenol oxidase, peroxidase, and sucrase, which aid in puncturing plant tissues digesting food, neutralizing secondary compounds and weakening plant defenses. To prey on nematodes, fungi with nematode-feeding behavior produce sticky webs, adhesive branches, and other specialized predatory organs through mycelial differentiation. Scavengers that use carrion as their primary energy source of their locus, such as vultures, bald storks, and hyenas, have evolved their specialized intake abilities to combat the many toxic energies found in carrion. The critical contaminants in carrion are toxins that bacteria produce. Botulinum toxin, which Clostridium botulinum produces under anaerobic conditions, is one of the most poisonous substances recognized by humankind. Large scavengers possess strong stomach acids, which is one reason why they can withstand microorganisms present in carrion. The scavenger's stomach acid strength is generally greater than that of humans, which acts as an inhibitory measure against germs, as the extremely acidic atmosphere suppresses the production of Clostridium botulinum spores, preventing the creation of botulinum toxin. Scavengers are protected from toxic energy damage due to specific antibodies. The serum of red-headed vultures and coyotes contains high levels of antibodies that can resist, bind, and eliminate botulinum toxin from the body. These antibodies are absent in humans, herbivores, and other non-scavenger species. Scavengers have several properties that can hinder bacterial growth in the gut. For instance, scavengers have higher levels of lysozyme in their digestive juices, and their food passes through their digestive tract faster. After choosing its food energy locus and its sublocus, a species must develop the biochemical reactions needed to ingest food, which is achieved through the secretion of catabolic enzymes associated with the energy locus. Digestive glands secrete catabolic enzymes that primarily break down molecules through hydrolysis. A species can develop a stable and advantageous feeding capacity at its energy locus using these specific catabolic enzymes. Animals belonging to different phyla or classes typically have significantly different catabolic enzymes to ingest food energy loci, in addition to variations caused by symbiotic relationships. Animals that have similar energy locus strategies also have similar functions of related digestive enzymes in their bodies. Pepsin, found in the gastric juice of higher animals, contains proteases with similar nature to those found in invertebrates. Chymotrypsin is commonly present in the pancreatic juice of higher animals. Amylase is abundant in the saliva and pancreatic juice of higher animals, as well as the digestive juices of lower animals. Xylanase, responsible for breaking down cellulose, is abundant in the intestinal fluids of herbivores like sheep and horses. Certain enzymes, like keratinase and penicillin acylase, can only be produced by microorganisms like fungi and actinomycetes.